My dear students, today we shall discuss about animal kingdom. If we can see around us, our environment, we can observe different type of animals. Very simple unicellular to multicellular and more complex organisms. The animal kingdom, initially the animal kingdom are divided into two large categories. One is invertebrate and second is vertebrate. Invertebrate, these are very simple. Their body starts from cellular organization, some organisms show tissue, some organisms show organ, and even the more advanced, some phyla show more advanced characteristics. These characteristics are system. Very early, primitive members are cellular. Then during the evolution, the cellular organization became more complex and complicated for tissue. Tissue in general case, generally cells are organized in two layers, outer ectoderm, inner endoderm. In the meantime, when the evolution goes on, more advanced organisms are developed. These are the organ system. In this case, they different type of organs, ectoderm, endoderm, mesoderms, coelom develop, and specific type of organs that arises. And ultimately, from annelid, the one of the non-chordate, to the modern mammals, different organs are organized to perform a specific function. They form the organ system organization. These are the invertebrate. And the vertebrate, main salient features of vertebrate, they possesses notochord and plus pharyngeal gill slit. These are the two major differentiation. Now, today's topic is the invertebrate phyla, their description and their specimen. When we consider invertebrate on the basis of their similarities, on the basis of different some characteristics, which characteristics on the basis of which invertebrate are classified? First characteristics is their body organization. Body organization. Second classification procedure is their coelom structure, coelom formation. Then the third point is their body symmetry. Body symmetry. And four, different type of system they develop, that is the respiratory, excretory, reproductive, even nervous. Considering all these facts, invertebrate are differentiated, are grouped into different phyla. These invertebrate phyla are, first is porifera, second Nidaria, third Ctenophora, fourth Platyhelminthes, fifth Nemathelminthes or Acoelominthes, Annelida, sixth is Annelida, seven Arthropoda, eight Mollusca, and ninth is Echinodermata. We shall discuss all the salient features that identifying characteristics of each phyla stepwise. First, we consider their first body organization, then coelom organization. What do you mean by body organization? Body organization may be simple or it may be complex. When cells are arranged in not a definite fashion, then this level is called tissue level organization. This is called first is tissue level or cellular level. Cellular level. Very simple form. These cellular simple form found in porifera. Then cells are organized. Suppose two layer of cells develop. Outer layer of cells called ectoderm and inner cell layer develop. This is called endoderm. Initially two derm, ectoderm and inner endoderm. This primitive structure generally is the simplest form and it is found in Nidaria and Ctenophora also. In between ectoderm and endoderm, some gelatinous substances may develop. This structure is called mesoglia. This is called mesoglia. Then, after this mesoglia formation, when evolution goes on, more complex character appear in their body organization. Then the structure comes. This structure is triploblastic. This is called as two layers is present here. This is called diploblastic. Then two layered structure converted into three layered structure. This is called the triploblastic. What are the triploblastic nature? Outer ectoderm. This is ectodermic layer, cell layers. Ectoderm cell layer. Inner cell layer is endoderm. This is endoderm cell layer. Blue color denotes in endoderm. And in between ecto and endo, another cell layers develop. This cell layer is called mesoderm. In this case, ecto, E for ecto, M for meso, and E for endo. three-layered structure development is triploblastic. This is called triploblastic. Simple cellular organization, unorganized cell. This organized cell, unorganized cell converted organized, forming two layer, ecto and endo. In between, mesoglia form. Then the three-layered triploblastic in nature body organization development. This triploblastic structure starts from Platyhelminthes, but during the course of evolution, this structure is not fixed. Gradually, in between the mesodermic area, some cells of the mesodermic area, suppose this is the ecto, this is the endo, and this is the total is the meso. This is ecto, this is endo, and these are meso. During the course of evolution, some intercellular, some gap, some pouch develops. That is, the mesodermic cells are differentiated into two layers, outer layer and inner layer. When mesoderm is differentiated into outer layer and inner layer, then a gap, then obviously a gap area originates. This gap area is called coelom. Then the coelomic structure start to develop. Initially, the true coelom initially not developed. True coelom, in this case, a continuous structure may not be developed in between ectoderm and endoderm. In this case, some scattered pouch originate within the mesoderm, and mesodermic cells surround it. That means this is the pseudocoelom. First, a solid triploblastic structure develop, then a pseudocoelom structure. This is acoelomate, no coelom. This is pseudocoelom arises. The pseudocoelom found in Acoelomate round one. Then the latter stage is pseudocoelom converted into true coelom. That is the one layer. This is called mesodermal outer layer and the inner mesodermal layer. Outer layer is ectoderm and inner layer is endoderm. A continuous layer develop between the two cellular layer of within that are present in the mesodermic zone. This is in this way true coelom originates. The true coelomic structure generally found in starts from Annelida to Echinodermata. This is the body organization and cellular and coelom formation. Next, the property is the body symmetry. What do you mean by body symmetry? Body symmetry, generally, when we divide the body in a vertical plane and it passes through the axis of the body, it can divide the body into two parts. When the vertical axis pass through the central point, then if the structures do not the same in their conformation, these are called asymmetry. When the vertical section pass through the central line at different angles, and when all these angles are all vertical lines pass through the central axis in all directions, and if in this case each segment is similar with the other segment, in this case this symmetry is called radial. But when the body is vertically divided by a plane that pass through the central axis and create only two same conformation, and this same conformation arise but in case of particular vertical section. When a single vertical section create two identical body structure, this is called the bilateral symmetry. The most primitive is the asymmetrical nature, then the radial, then the most advanced is bilateral. What do you mean by radial and different in the body symmetry are three types: asymmetry, radial, bilateral. What is asymmetry? When the body, this type of body, when we dissect this body in different angle, we do not get any piece that is similar with the other. All different species are dissimilar with each other. There are no symmetry. That is why these are called asymmetrical. This is found in porifera. Porifera is asymmetrical. But radial symmetry. What do you mean by radial? When a body is divided by any vertical plane that pass through the central axis, then the part that develop and each part is similar with the other part, then this type of symmetry is called radial symmetry. What is the radial symmetry? Suppose we draw a structure. Then when we cut this body part, when we cut this body part and this section pass through the central axis, in this case the two plane, here we draw two plane. In this case, when we body dissect in such two plane and each part resemble with the other part, if the structure is properly radial in structure, this is the radial symmetry. But the bilateral symmetry, this radial symmetry we found in the bilateral symmetry developed next to the class. This starts from platy to echino. What is bilateral symmetry? In the bilateral symmetry, suppose this structure. When we cut this body in this plane, two half develop. If we cut this plane in this way or in this way, we do not get the similar body structure. Only when the vertical line pass to the central axis, only in one specific direction, two half develop, and these two half are mirror image with each other. In other cases, this will not be happen. This is the bilateral symmetry. Bilateral symmetry is the most advanced form. Then this is the total outline of classification of invertebrate. Here a term is very important. Invertebrate, that means vertebral column is totally absent. What is the source of the vertebral column? Vertebral column originates from the notochord. So, in case of invertebrate, notochords are totally absent. Notochord is totally absent. In place of notochord, the higher members show some development of neural nervous system. These nervous systems may be ganglia or it may be paired, ventral, solid nerve cord. These are the more or less salient features of invertebrate. In the later class, I will discuss different salient characteristics: Porifera, Nidaria, Ctenophora, Platyhelminthes, Acoelomate, Annelida, Arthropoda, Mollusca and Echinodermata in section wise.